Close your eyes and come with me. We're travelling 4.5 billion years backward to a time when stellar nurseries gave birth to wandering worlds, when a distant sun breathed its dying light into the endless dark. In that ancient exhalation, wrapped in crystalline ice and organic molecules no larger than specks of dust, something waits, not thinking, not dreaming, but alive in the deepest sense. Microbial life, its biological machinery frozen between existence and void, beginning an odyssey across the galaxy. Hello there, and welcome to Earth and Beyond, where planetary wonders drift into cosmic dreams, and science takes the long way home. Tonight, we're following an extraordinary visitor that swept past Mars just months ago, carrying within its primordial ice what might be the most significant biological event in our planet's recent history. This isn't speculation. It's a real interstellar wanderer called 3I Atlas, and the microscopic passengers it may have carried could be rewriting the story of life in our solar system right now. Imagine an object no larger than a city block, frozen for ages beyond measurement, travelling through the absolute silence between stars. As it approaches our sun, something extraordinary happens. The ice begins to sublime, releasing not just vapour and debris, but perhaps something far more precious. The preserved essence of life from another world. Microorganisms that have waited eons for this moment of potential awakening. The mathematics of this cosmic delivery are both overwhelming and beautiful. As this wanderer shed material at rates measured in hundreds of grams per second, it created an enormous cloud of particles, each one a potential vessel carrying biological information across the void. Some of these particles drifted toward Mars, others may find their way to Earth, and if even a small fraction contains viable organisms, we may be witnessing the most profound biological event in recent cosmic history. Before we drift deeper into this mystery, I'd like to know, where in this vast cosmos are you listening from tonight? As you settle into this exploration, consider that the very atoms in your body were forged in stellar furnaces, and perhaps some of the microorganisms in your system trace their ancestry to deliveries from distant stars. If you find this kind of gentle cosmic exploration soothing, I'd be grateful if you gave the video a like, or perhaps subscribed. It's a small gesture, but it helps this quiet space reach others who might also want to wander among the mysteries that connect worlds. I'm not asking because numbers matter here in the usual way. I'm asking because the more people who join these contemplations, the richer our shared wandering becomes. So close your eyes if you haven't already. Let your breathing slow to match the rhythm of distant starlight, and let's drift beyond the familiar into realms where biology meets cosmology. October 19th, 2019. A routine sky survey picks up something unusual. A fuzzy object moving against the background stars, following a trajectory that suggests an origin far beyond our solar system. What astronomers have discovered will challenge everything we thought we knew about the isolation of planetary systems, and the ways life might journey between worlds. The cosmic seed we're following tonight isn't just a single microorganism waiting in the ice, it's an entire philosophy of existence. The idea that life does not respect the boundaries we draw around solar systems, that bacteria can exercise patience beyond human comprehension, and that the universe may be far more alive than we ever dared contemplate. Consider what it means for a bacterium to persist in interstellar space. The conditions are beyond extreme. Temperatures hovering just above absolute zero. Radiation that would shatter deoxyribonucleic acid in moments. And a void so complete that a living cell might wait millions of years between encounters with a single photon. Yet research reveals something that sounds impossible. Until you examine the evidence, certain bacteria can not only survive these conditions, but enter a state so close to death that the distinction becomes philosophical. In laboratories around the world, scientists have been testing the boundaries of bacterial endurance. They have exposed microorganisms to radiation doses thousands of times greater than what would kill complex life. They have frozen them at temperatures that halt all chemical reactions. They have placed them in vacuum chambers that simulate the emptiness between stars, and again, they have discovered that life finds ways to persist. that challenge our understanding of what persistence means. Take Deinococcus radiodurans, a bacterium so resilient scientists nicknamed it Conan the Bacterium. When researchers bombarded it with gamma rays at doses that would sterilize medical equipment, the organism did not just survive, it flourished. After receiving radiation levels 28,000 times greater than what would kill a human, these bacteria repaired their shattered deoxyribonucleic acid and continued reproducing as if nothing had happened. The mechanism is elegant in its simplicity. These organisms maintain multiple copies of their entire genome, scattered throughout the cell like backup drives in a cosmic data center. When radiation destroys one copy, specialized enzymes scan the wreckage, identify the damage, and rebuild the genetic code using information from the intact copies. It is not perfect. Some errors creep in, mutations that become the raw material for evolution, but it works well enough to preserve life across timescales that make geological ages seem brief. This is the foundation of our cosmic seed hypothesis. If bacteria can survive such punishment on Earth, what might they endure during a journey between stars? The answer, according to recent research, is both longer and more severe than anyone expected. Bacterial spores embedded in ice and shielded from direct radiation exposure could remain viable for hundreds of millions of years, even under the harsh conditions of interstellar space. But survival is only the first challenge. The cosmic seed must also travel. And here we encounter one of the universe's most patient transportation systems. Comets are not the simple ice balls of popular imagination. They are complex structures containing chambers and layers of different materials, some dating back to the formation of their parent solar systems, in the coldest regions of these wandering islands, far from any warming influence. Bacterial life could remain in perfect suspension, waiting. 3i Atlas represents exactly this kind of vessel. When astronomers first analysed its composition, they found it unlike any comet from our solar system. Its chemistry suggested an origin around a red dwarf star, probably in a system where conditions favoured the preservation of organic molecules. The ratio of carbon monoxide to water in its coma indicated formation at temperatures even colder than our Kuiper belt, creating ideal conditions for long-term biological preservation. As this ancient wanderer approached our sun, something remarkable began to happen. The heat of our star started to sublime the ice from its surface, creating a coma of gas and dust that stretched for thousands of kilometers. But this was not just a celestial light show. It was potentially the opening of our biological time capsule that had been sealed since before Earth's complex life evolved. The material streaming away from 3i Atlas included particles ranging from microscopic dust grains to fragments the size of small stones. Each particle followed its own trajectory, some accelerated by solar radiation pressure, others influenced by the gravitational pull of planets. The result was a vast, expanding cloud of debris that would eventually span millions of kilometers, intersecting the orbits of multiple worlds. Here is where our story becomes deeply personal. As this cloud expanded, some portion of it crossed paths with Mars at precisely the right moment. The red planet was positioned in its orbit like a cosmic catcher's mitt, ready to receive whatever gifts this interstellar visitor might offer. The geometry was not perfect. 3i Atlas passed Mars at a distance of roughly 29 million kilometers, but when you're dealing with a debris cloud that large, even such vast distances become manageable. The particles that reached Mars were not travelling alone through hard vacuum. Many were embedded in large aggregates, or ice crystals that provided some protection from the worst of the cosmic radiation. The journey from the comet to Mars would have taken weeks or months, during which time the biological cargo would face its greatest test, survival in the open space between worlds. But Mars is not like Earth. Our planet's thick atmosphere creates a furnace for incoming particles, burning up most meteors in brilliant streaks of light. Mars, with its whisper-thin atmosphere, offers a gentler reception. Particles that would be vaporized before reaching Earth's surface can drift down onto Mars almost intact, settling onto the dusty plains like cosmic snow. If bacteria from 3i Atlas survived the ejection from their icy home, if they endured the radiation and cold of their journey through space, if they remained viable during their descent through Mars' tenuous atmosphere, Then they would face their final challenge, awakening in a world unlike any they had known before. The surface of Mars is hostile beyond imagination. Ultraviolet radiation bathes the landscape, breaking down organic molecules within hours. Pervasive chemicals called perchlorates poison the soil, creating conditions toxic to most forms of life. Temperatures swing from barely above freezing to colder than Antarctica's worst winter, sometimes in the span of a single Martian day. But bacteria are resourceful in ways that constantly surprise researchers. Many species can enter a state called cryptobiosis, a complete suspension of biological activity that allows them to survive conditions that would destroy active cells. In this state, they are neither alive nor dead in any meaningful sense. They are waiting. And here is the remarkable thing about Mars. Beneath its hostile surface lies a different world entirely, a few meters down. Shielded from radiation and buffered from temperature extremes, the subsurface offers conditions that might actually nurture life. Ice deposits provide water, volcanic heat creates pockets of warmth, minerals supply the chemical energy that bacteria need to power their metabolism. Recent discoveries have mapped extensive networks of subsurface ice throughout Mars' mid-latitudes. Ground-penetrating radar has detected what appear to be liquid water lakes beneath the polar ice caps. Geothermal activity, while limited, creates zones where temperatures rise high enough for bacterial metabolism to function, albeit slowly. If bacteria from 3I Atlas reached these protected environments, they would not need to compete with the thriving biosphere as they would on Earth. Mars may once have hosted life, but if so, that life either vanished long ago or retreated to isolated refuges deep underground. The newcomers might find a world of vacant ecological niches, ready for colonization. This is the cosmic seed hypothesis in its full scope. Not just the idea that life can travel between stars, but the possibility that it does so regularly, seeding worlds throughout the galaxy with biological potential. Every comet that ventures into the inner solar system becomes a potential ark. Every close encounter between an interstellar visitor and a planet represents a moment when evolution might leap forward by millions of years. The implications stretch far beyond Mars. As 3I Atlas continued its journey through our solar system, its trajectory carried it past Earth's orbital path. The distance was much greater, over 270 million kilometers at closest approach. But the same principle applies. Material ejected from the comet spreads out in expanding sphere, and some infinitesimal fraction of that material will eventually intersect Earth's orbit. Most will burn up in our atmosphere, our planet's robust defense against space debris, but not all. Scientists studying meteor showers have long known that fine dust from comets reaches Earth's surface constantly. About 100 tons per day of extraterrestrial material drifts down through our atmosphere, most of it invisible, unnoticed, becoming part of the soil we walk on and the air we breathe. The smallest particles, those in the micron range, do not heat up enough during atmospheric entry to sterilize their contents. They fall gently, taking weeks or months to complete their descent, protected by Earth's atmosphere from the worst of the cosmic radiation they encountered in space. If some of these particles originated from 3I Atlas, if some contain bacterial material from another star system, then we may already be living alongside visitors from distant worlds without knowing it. This possibility transforms how we think about life on Earth. We are accustomed to viewing our planet as an isolated biological experiment, evolution proceeding in splendid solitude over billions of years. But if the cosmic seed hypothesis is correct, Earth may have been receiving regular deliveries of genetic information throughout its history. Our biosphere might be a hybrid, carrying biological innovations from multiple star systems, blended together through eons of cosmic exchange. The bacteria in your gut, the microorganisms in the soil, the vast communities of microbes that drive Earth's biochemical cycles, all of them might carry genes that originated elsewhere, so thoroughly integrated into terrestrial life that we cannot distinguish them from homegrown innovations. We may not just be products of Earth's evolution, we may be products of galactic evolution, shaped by biological information that has flowed between worlds for as long as life has existed. This is the profound possibility we explore tonight. that life recognizes no boundaries between worlds, that genetic information travels the galaxy on patient messengers made of ice and rock, and that every world capable of nurturing life eventually receives visits from the cosmos's vast biological library. The cosmic seed from 3i Atlas may have already planted itself in Martian ice, beginning the slow process of adaptation to a new world. It may have reached Earth, adding new capabilities to our planet's already enormous microbial diversity. Or it may have failed entirely, joining the countless other attempts at interstellar colonization that did not quite succeed. But succeed or fail, it represents something magnificent. Life's relentless drive to persist, to spread, to find every possible niche where existence is possible. It is a reminder that we live in a universe where biology and cosmology intersect, where the patient mechanisms of evolution operate on scales that encompass entire galaxies, and where every comet carries the potential for worlds to share their most precious gift, the spark of life itself. As we drift toward our next segment, hold this thought. You may be carrying within your own cells the genetic legacy of bacteria that travelled between stars, organisms that waited billions of years to contribute to the magnificent complexity that is terrestrial life. You're not just human, you're a cosmic hybrid, a walking example of life's ability to transcend the boundaries between worlds. We return now to the moment when two worlds' histories converged, not through collision, but through something far more subtle and profound. Picture this: October 12th, 2019, when 3i Atlas swept past Mars at a distance that, in cosmic terms, might as well have been a whisper between old friends. 29 million kilometers separated the comet from the red planet, a gap that sounds impossibly vast until you consider that this interstellar visitor was shedding material like a cosmic dandelion in a stellar wind. The geometry of this encounter was not random. Mars, in its endless dance around the Sun, found itself positioned precisely where 3i Atlas's debris trail would intersect its orbital path. Some scientists have calculated that with a velocity adjustment of merely 8 to 12 kilometers per second during its approach, the comet could have achieved a direct collision trajectory. It did not. But the precision of this flyby raises questions that stretch beyond simple orbital mechanics into the realm of cosmic choreography. What matters for our story is not whether this encounter was guided or coincidental. What matters is understanding what happened to all that material the comet was releasing into space. Remember, 3i Atlas was losing between 100 grams and 800 grams of mass every single second as it approached perihelion. That is not a gentle dusting of space. That is a constant torrent of particles ranging from the microscopic to fragments the size of pebbles, each one following its own slightly different trajectory based on when and where it separated from the comet's surface. Mars Reconnaissance Orbiter, Mars Express, and the European ExoMars Trace Gas Orbiter all turned their instruments toward the comet during this historic flyby. They captured images showing a coma several thousand kilometers across, though the exact boundaries proved difficult to measure because the brightness faded gradually into the background noise of space. What they could not detect, because the resolution simply was not sufficient, were the individual particles, the dust grains in the micron to millimeter range that would be most likely to carry intact biological material. Here is what we understand about how particles behave when a comet passes near a planet. They do not all continue along the comet's original path like obedient soldiers in formation. Gravity, even Mars's relatively modest gravitational field, tugs at them with invisible fingers. Solar radiation pressure pushes them like an ethereal wind. The particles spread out in a complex pattern that resembles nothing so much as seeds dispersing from a cosmic flower. Some accelerate ahead of the comet, some lag behind, some deflect toward the planet. The smallest particles, those in the micron range where bacterial spores might hide, are most affected by radiation pressure and can be blown into trajectories that intersect planetary orbits with almost surgical precision. It is a phenomenon astronomers call orbital resonance, the same gravitational dance that keeps Jupiter's moons in their eternal waltz. Now applied to microscopic debris, carrying perhaps the most precious cargo in the universe. Multiple spacecraft were monitoring Mars for exactly this kind of event during the days surrounding the flyby. The rovers on the surface, Perseverance and Curiosity, were both tasked with watching for any unusual atmospheric phenomena during the critical period. They observed nothing dramatic, no visible meteor showers or sudden dust storms that could be attributed to the comet's debris. But the absence of spectacle does not mean nothing reached the surface. Most particles small enough to carry protected bacteria would be too tiny to create visible meteors. They would simply drift down through Mars' gossamer atmosphere, taking days or weeks to settle through the thin carbon dioxide that barely qualifies as an atmospheric envelope. It is a gentle descent, more like falling through honey than plummeting through Earth's robust atmospheric furnace. Let us walk through the sequence of events that a bacterium would experience if it survived this cosmic odyssey. First, imagine being frozen inside 3I Atlas's ice matrix for billions of years, your cellular machinery completely suspended in a state that is neither life nor death but something in between. No metabolic activity whatsoever, just the essential architecture of existence preserved in crystalline perfection. As 3I Atlas approaches the Sun, portions of the comet heat up and begin sublimating, turning ice directly into gas with the violence of controlled explosions. This process creates jets of escaping vapor carrying dust and larger fragments at velocities that can exceed 300 meters per second. For most organisms, this would mean instant destruction. The acceleration alone would tear cellular structures apart like tissue. Paper in a hurricane, but bacterial spores are different. They are evolution's answer to exactly this kind of mechanical stress. Studies have shown that bacterial endospores can withstand accelerations of hundreds of thousands of times Earth's gravity without losing viability. The pressures involved in cometary outgassing, while extreme, fall well within their tolerance range. So step 1, ejection from the comet, is survivable for the right kind of organism. Step 2 involves the transit through space from 3I Atlas to Mars, a journey that would take anywhere from days to weeks, depending on the particle's exact trajectory and velocity. During this time, cosmic radiation becomes the dominant threat. These high-energy particles constantly bombard everything in space, breaking chemical bonds and damaging deoxyribonucleic acid with the patience of cosmic sculptors working in destruction. But remember, these particles are not travelling alone through their hard vacuum. Many are embedded in larger dust aggregates or ice crystals that provide some shielding from the worst radiation. It is like travelling in a cosmic submarine, where layers of ice and rock provide protection from the lethal environment outside. The journey from 3I Atlas to Mars represents significant radiation exposure, but not necessarily lethal doses, especially for organisms with deoxyribonucleic acid repair mechanisms as robust as those found in extremophiles. Step 3 brings us to entry into Mars' atmosphere, where Earth bacteria would face their greatest challenge. But Mars's atmospheric envelope is only about 0.8% the density of Earth's. Entry heating is minimal for small particles. Something the size of a grain of sand would barely warm up at all during its leisurely descent. It would flutter down through the thin carbon dioxide atmosphere like a feather through near vacuum, taking its time, gradually settling toward the surface with none of the drama we associate with meteor impacts. No fireball, no plasma sheath, no violent friction heating, just a gentle descent through an atmosphere so tenuous it might as well not exist. This is radically different from Earth, where our thick atmospheric blanket creates a furnace that incinerates most cosmic visitors before they can reach the ground. Step 4 involves landing, and where a particle touches down on Mars determines everything that follows. If it hits bare rock in the middle of a Martian day, when surface temperatures can briefly reach above freezing, ultraviolet radiation would sterilize it within hours. Mars lacks the protective ozone layer that shields Earth's surface, leaving the landscape bathed in ultraviolet light that would destroy any organic molecules exposed to direct illumination. But if the particle lands in shadow, if it settles into a crack or crater where dust can cover it, if it happens to drift down in one of the polar regions where ice deposits accumulate throughout the Martian year, then survival becomes not just possible, but probable. The difference between life and death on Mars often comes down to luck. being in the right place, when cosmic chance decides your fate. The research on Martian ice deposits is particularly relevant here. Scientists have mapped extensive ice formations at both Martian poles and in mid-latitude regions, where seasonal cycles create alternating layers of water ice and frozen carbon dioxide. Some of these deposits are pure water ice, crystalline and protective. Others are mixed with regolith, the soil and rock material that covers the planet's surface like a rusty blanket. Studies on bacterial survival in different types of ice show dramatically different results, and these differences could mean everything for our cosmic passengers. In pure water ice under Mars-like conditions, bacteria can survive for tens of millions of years, their cellular machinery preserved like biological amber. In ice mixed with Martian soil, survival times drop to hundreds of thousands of years due to chemical interactions between the ice, soil minerals, and radiation-damaged organic molecules. But here is what makes the 3I Atlas scenario so compelling. The particles settling onto Mars would not need to survive for millions of years in their initial landing location. They would only need to persist long enough to reach a more protected environment, and Mars offers numerous pathways for such migration. On Earth, we have discovered that bacteria can move through soil and rock layers, either by growing along moisture gradients or by being transported in flowing water. Mars does not have surface water flows now, but it does have ice, and where there is ice, especially in regions with minimal geothermal heat, there is the possibility of liquid water existing in thin films between ice grains or in larger pockets deeper underground. Recent research has identified the most likely locations for liquid water on Mars, and the findings are remarkable. Subsurface lakes have been detected beneath the southern polar ice cap using ground-penetrating radar. These are not lakes in the familiar Earth sense. They are likely brine solutions with high concentrations of dissolved salts that keep them liquid at temperatures well below 0 degrees Celsius. But liquid is liquid, and for bacteria, that represents possibility. The Perseverance rover has been examining rocks from an ancient river delta in Jezero crater, and in recent months, the science team published findings on rock formations they call Bright Angel. These rocks are fine-grained mudstones rich in oxidized iron, phosphorus, sulfur, and organic carbon, a chemical combination that on Earth typically results from very specific processes. The patterns in these rocks show features that, on our planet, are only produced in two ways, either through purely chemical processes at high temperatures or through biological activity, where bacteria use chemical reactions between iron, sulfur, and organic matter as an energy source. The crucial detail is temperature. All analyses of these Martian rocks indicate they were never heated above about 75 degrees Celsius. But the chemical processes that could create these features without biology require much higher temperatures, several hundred degrees at minimum. If the rocks were never that hot, and they contain these specific patterns of minerals and organic compounds, then biology becomes the most plausible explanation. Not proof, certainly. Science does not work that way. But the most consistent hypothesis given the available data suggests that life processes may have shaped these formations. Now connect this to our 3I Atlas scenario. If bacteria from the comet reached Mars and made their way into subsurface environments where liquid water exists, they would encounter conditions remarkably similar to what generated those patterns in the Bright Angel rocks. Iron and sulfur are abundant in Martian regolith. Organic carbon might be sparse on the surface, but deeper down, protected from ultraviolet radiation, organic molecules from meteorites and from any native Martian biological activity could persist for geological ages. The metabolic pathways that bacteria use to extract energy from iron and sulfur reactions are exactly the kind of universal biochemistry that would work anywhere in the solar system. These reactions do not depend on sunlight or complex organic nutrients, just simple chemical gradients between different compounds. It is the most basic form of a life imaginable, and possibly the most resilient. There is another piece to this puzzle that has only recently come into focus, and it involves what scientists call microtunnels. These are tiny channels in Martian rocks ranging from a few micrometers to tens of micrometers in diameter. They twist and curve through the mineral matrix in patterns that look remarkably similar to channels created by bacteria on Earth. When they dissolve rock to extract nutrients, the morphology matches Earth examples almost perfectly. The size distribution matches. The spatial patterns match. What we cannot prove yet is whether those tunnels were created by biological activity or by some purely chemical process we have not identified. But if bacteria from 3I Atlas established themselves in Martian subsurface environments, these microtunnels could represent their signatures, evidence of biological activity that occurred not billions of years ago, but months ago. The organisms would not need to be alive today for us to detect their influence. They could have thrived for a few years, decades, or centuries after arriving, then died out when conditions changed or resources were exhausted. But they would leave traces, microtunnels in rocks, unusual concentrations of specific isotopes, molecular fossils, fragments of cellular structures that resist degradation. This is what makes our timeline different from previous speculation about Martian life. We are not talking about ancient Mars billions of years ago, when the planet had a thick atmosphere and liquid water on the surface. We are talking about now. We are talking about an injection of biological material that happened within living memory, an event so recent that the organisms involved might still be adapting to their new home. The adaptation process, if it is occurring, would be brutal and swift. Most organisms would die immediately upon exposure to Martian conditions. A few might survive in a dormant state, waiting in cryptobiosis for better conditions to return. An even smaller number might find themselves in environments where they can actually metabolize, grow, and reproduce slowly. Those survivors are the ones that matter for evolution. They are the ones that would begin the process of becoming Martian, rather than merely visitors to Mars. The selective pressures would be intense beyond anything we see on Earth. Water availability would be the first constraint. Any bacterium that could not extract water from ice, or tolerate extremely high salt concentrations, would simply cease to exist. Radiation resistance would be the second challenge, even underground. Cosmic rays and radioactive decay from Martian rocks create a constant background of ionizing radiation. The organisms would need robust deoxyribonucleic acid repair mechanisms, or they would accumulate mutations faster than they could reproduce. Energy sources would be the third constraint. There is no sunlight underground, no organic matter raining down from a biosphere above. The only energy available comes from chemical reactions between minerals, and the extremely limited dissolved gases in whatever liquid water exists. It is an energy budget so tight it makes Earth's deep subsurface bacteria look pampered. But bacteria are survivors in ways that constantly astonish researchers. They have colonized every environment on Earth, from kilometers deep in the continental crust to the upper atmosphere. They have adapted to conditions we keep thinking should be impossible. Boiling acid, pure salt, solid rock, hard vacuum. If bacteria from 3I Atlas carried the right genetic toolkit, if they had genes for radiation resistance and for extracting water from minerals, for metabolizing simple chemical reactions, then Mars could become their home. And here is the truly fascinating possibility. If bacteria from 3I Atlas did successfully colonize Martian subsurface environments, we might never detect them with current technology. Our rovers can only drill a few meters down. The planned sample return missions are designed for surface materials. The ice deposits where life is most likely to survive are buried beneath meters to kilometers of rock and regolith. We would need to mount a dedicated mission specifically designed to reach those depths, extract samples from subsurface ice deposits, and analyze them for biological signatures using techniques sensitive enough to detect sparse microbial populations. That kind of mission is probably decades away, which means that right now, as we orbit Mars with our spacecraft and roll across its surface with our rovers, there could be an entire ecosystem developing beneath our sensors. Not native Martian life that evolved over billions of years, not ancient fossils waiting to be discovered, but a new biosphere born from an interstellar delivery just months ago. The bacteria would be dividing, mutating, adapting to conditions their ancestors never encountered. In a few generations, they would not look anything like their progenitors from 3I Atlas's home system. They would be Martian bacteria, optimized for Martian conditions, carrying genetic information from another star, but shaped by the red planet's unique environment. The encounter between 3i Atlas and Mars was not just an astronomical event that we observed and catalogued. It was potentially a biological event whose consequences are still unfolding in hidden chambers beneath the Martian surface. What grows from that encounter, if anything does, remains invisible to our current instruments, but might represent the most significant development in our solar system's biological history. As we drift toward our next exploration, consider this. The boundary between worlds is not as absolute as we once believed. Bacteria do not recognize the lines we draw on star charts. They do not respect the vast distances that separate planets. Given patience, protection, and just a little bit of cosmic luck, life finds ways to cross the voids that seem impossible to bridge. The encounter we have witnessed may have planted seeds that will grow into entire ecosystems, hidden from view, but slowly transforming Mars from a dead world into a living one. And if that is happening, if Martian bacteria are even now dividing in subsurface ice deposits, then we are witnessing something unprecedented, the birth of a new biosphere, delivered by Cosmic Express from worlds we will never see. Let us talk about what it actually takes to survive in the cosmic arena. Not the sanitized version with protective spacesuits and life support systems, but the raw, unforgiving reality of hard vacuum, cosmic radiation, and temperatures that oscillate between barely above absolute zero and scorching heat, depending on whether you are bathed in starlight or hidden in shadow. Because if we are going to seriously consider bacteria journeying aboard 3i Atlas, we need to understand the actual physics and biology of survival at the extremes. Conan the bacterium earned its nickname through feats that sound like science fiction until you examine the data. When researchers exposed Deinococcus radiodurans to radiation doses that would kill a human 28,000 times over, the organism did not just survive, it flourished. After receiving gamma-ray doses totaling 150,000 Gray, about 30 times what would completely sterilize medical equipment, the bacteria repaired their shattered deoxyribonucleic acid and continued reproducing as if the apocalyptic radiation had been nothing more than a gentle summer breeze. The mechanism behind this resilience is both elegant and profound. These organisms maintain multiple copies of their entire genome, scattered throughout the cell like redundant backup systems in a cosmic data center. They also possess extraordinarily efficient deoxyribonucleic acid repair enzymes that can essentially rebuild the genetic code from fragments. It is not reconstruction. It is resurrection at the molecular level. Life rebuilding itself from the wreckage of near destruction. But radiation resistance represents just one variable in the survival equation. Temperature poses an equally formidable challenge. And here we encounter conditions that stretch the very definition of what constitutes a habitable environment. In the shadow of an interstellar object travelling through space, temperatures hover around minus 270 degrees Celsius, just 3 degrees above absolute zero, the point at which all molecular motion essentially ceases. At these temperatures, water ice becomes harder than steel. Proteins cannot fold or unfold. Chemical reactions stop entirely. For most organisms, this represents not just death, but complete molecular stillness. A state where life processes become physically impossible. For bacterial spores, however, it is something entirely different. It is preservation. The spore form represents one of evolution's most brilliant solutions to hostile environments. When conditions deteriorate beyond tolerance, certain bacteria do not merely slow their metabolism, they dismantle it entirely. The spore encases its deoxyribonucleic acid with protective proteins and surrounds everything in multiple layers of specialized membranes and thick protective coats. Then it purges almost all its water content, creating a desiccated capsule where biochemistry cannot occur because there is no liquid medium for reactions to proceed. In this state, the organism exists in a liminal realm between life and death. It is not alive in any meaningful sense. There is no metabolism, no growth, no response to stimuli. But it is not dead either. It is waiting. Sometimes for periods that make geological ages seem brief. International Space Station experiments have proven that this concept works in actual space conditions. Researchers mounted bacterial cultures on the station's exterior, directly exposing them to vacuum, radiation, temperature extremes, and solar ultraviolet light. After 3 years of this cosmic punishment, they retrieved the samples and found something remarkable. Some bacteria remained viable, not just chemically intact, but capable of reviving when given water and nutrients. The survival rate was not high. Perhaps 1 cell in 10 million retained viability. But that is precisely the point. Evolution does not require high survival rates. It requires only that some survivors exist. And when you consider the scale involved in cometary panspermia, even these seemingly impossible odds become manageable. 3I Atlas has a diameter of approximately 500 meters, giving it a volume of roughly 65 million cubic meters if we assume a roughly spherical shape. If even a tiny fraction of that volume contained bacterial spores at concentrations found in Earth's soil, about 1 billion spores per cubic centimeter, we would be talking about numbers that exceed human comprehension. When the comet shed material near Mars, even if only one particle in 1 billion contained viable bacteria, and even if only one bacterium in 1 million from those particles survived the journey and landing, thousands of organisms could still reach the Martian surface alive. But survival is not just about the journey, it is about what happens after arrival. This is where the specific conditions on Mars become absolutely critical to our story. The planet's surface is hostile, but that hostility is not uniform across all locations and depths. Radiation levels vary dramatically depending on geography and on how deep you go underground. At the surface in equatorial regions, the radiation dose averages about 80 milligrays per year, roughly equivalent to receiving 35 full-body computed tomography scans annually. That is sufficient to prevent most life as we know it from surviving long-term exposure. But descend just 2 meters below the surface, and you are behind enough shielding to reduce radiation exposure by 95% or more. The subsurface becomes a different world entirely. Mars's temperature profile follows a similar pattern of dramatic variation. Surface temperatures can briefly exceed 0 degrees Celsius at the equator during summer afternoons, but they plunge to minus 130 degrees Celsius at night. This daily thermal shock would be lethal to most organisms. The subsurface, however, remains thermally stable, insulated from these wild swings by meters of rock and soil. In regions with even minimal geothermal activity, heat flow from the planet's interior can maintain subsurface temperatures where liquid water becomes possible if the right dissolved salts are present. Perchlorate salts are abundant in Martian soil, and they represent both a hazard and an opportunity for bacterial life. In high concentrations, perchlorates are toxic, oxidizing cellular structures and disrupting normal metabolism. But some Earth bacteria have evolved not only to tolerate perchlorates, but to use them as an energy source. Certain species can metabolize perchlorate compounds, breaking them down and using the released oxygen for respiration even in environments completely devoid of gaseous oxygen. If bacteria from 3I Atlas carried similar capabilities, or if they could acquire those genes through horizontal transfer from any native Martian organisms, perchlorate-rich environments would transform from barriers into habitats. The very chemistry that makes Mars seem hostile to life could become the foundation for life's persistence. The energy budget for subsurface bacterial life on Mars would be austere but sustainable. On Earth, bacteria living kilometers deep in rock extract energy from the oxidation of hydrogen gas produced by chemical reactions between water and iron minerals. The process is extraordinarily slow. These bacteria can take centuries to complete a single reproductive cycle, making them the slowest growing organisms known to science. But slow growth is still growth. It is still evolution in action. It is still life continuing against overwhelming odds. Mars possesses abundant iron minerals. The planet's rusty appearance comes from iron oxide covering nearly every surface. Deeper down, unoxidized iron reacts with available water to produce hydrogen gas. That hydrogen becomes biological fuel for organisms capable of capturing it. Bacteria that can harness this hydrogen and use it to reduce carbon dioxide into organic molecules possess an energy source that does not depend on sunlight or organic nutrients from above. They become primary producers in ecosystems with exactly one member: themselves. They create food from nothing but chemistry, the most fundamental form of life imaginable. laboratory simulations of Martian subsurface conditions have yielded surprising results that challenge our assumptions about life's limits. When researchers create environments matching Mars' temperature, pressure, mineral composition, and radiation levels, then introduce various bacterial species. Some do not just survive. They adapt in real time. Over hundreds of generations in these punishing conditions, the bacteria evolve. Mutations that would be neutral or harmful in Earth conditions become beneficial under Martian constraints. Genes that were rarely expressed suddenly become critical for survival. The organisms do not become simpler in response to stress. They become differently complex, optimized for a completely new set of environmental challenges. One particularly revealing study examined bacterial metabolism under simulated Martian atmospheric pressure and temperature. At 6 millibars of pressure, about 0.6% of Earth's sea level pressure, and at temperatures just above freezing, only one bacterial species out of 30 tested showed growth. That species, Serratia liquefaciens, managed to reproduce despite conditions that halted all its relatives. The key was its ability to maintain cellular pressure gradients, even when external pressure dropped below what most organisms can tolerate. A single metabolic adaptation, probably controlled by just a handful of genes, made the difference between death and survival. This is exactly why horizontal gene transfer becomes so crucial in our scenario. If bacteria from 3I Atlas landed on Mars carrying biochemistry completely alien to terrestrial conditions, they would almost certainly die. The mismatch between their genetic programming and Martian reality would prove insurmountable. But if those organisms carry genes that are functionally similar to what Earth bacteria use, just optimized for different conditions, then gene exchange becomes possible. A Martian bacterium, if any exist, could acquire radiation resistance genes from the newcomers. An alien bacterium could pick up pressure tolerance genes from a native Martian survivor. Both lineages benefit from the exchange. Both increase their chances of long-term survival. The mechanism for gene transfer does not require direct cell contact as it often does on Earth. In extremely nutrient-poor environments, bacteria release deoxyribonucleic acid fragments when they die. Those fragments can persist in the environment, especially in ice or mineral matrices, for thousands of years. When a living cell encounters those fragments, it can sometimes incorporate them directly into its own genome through a process called transformation. It is the same mechanism by which antibiotic resistance spreads through bacterial populations, but operating in the Martian subsurface rather than in hospitals. What works in clinical settings can work in ice deposits millions of kilometers away. There is another factor that makes Mars potentially more hospitable than the initial impressions suggest. The planet's low gravity, about 38% of Earth's, means bacterial cells experience less physical stress. On Earth, bacteria in deep subsurface soil must cope with enormous pressures from overlying rock. On Mars, the same depth results in much lower pressure, meaning cellular structures do not need to be as robust. Energy does not have to be diverted to maintaining structural integrity against crushing force. That is metabolic energy that can be used for growth and reproduction instead, a crucial advantage in an environment where every calorie must be carefully conserved. The water situation on Mars proves more complex than simple statements about liquid water's impossibility on the surface. While pure water cannot remain liquid under Martian atmospheric conditions, salt solutions behave very differently. Perchlorate brines can remain liquid at temperatures as low as minus 70 degrees Celsius. At those temperatures, metabolism would be incredibly slow. Enzymes would function at rates thousands of times slower than in warm water, but slow is not zero. For organisms with lifespans measured in millennia, such conditions remain acceptable. Recent discoveries have identified specific locations on Mars where these conditions seem optimized for microbial survival. The recurrent slope lineae, dark streaks that appear seasonally on certain Martian slopes, might represent brine flows. Radar detection of subsurface liquid water beneath the south polar ice cap suggests entire lakes of salty water existing in perpetual darkness. Gullies and crater walls show evidence of recent fluid flow, possibly from eruptions of subsurface aquifers. Each of these environments could harbour life if organisms could reach them, and adapt accordingly. The survival equation ultimately reduces to several key variables: radiation shielding, solved by moving underground; temperature management, solved by finding geothermally influenced regions or accepting extremely slow metabolism; water access, solved by utilizing brines that remain liquid below freezing; energy sources, solved by chemosynthesis using minerals and simple gases; genetic flexibility, solved by horizontal gene transfer allowing rapid adaptation. Every variable in this equation has solutions demonstrated by Earth organisms. Every solution involves biochemistry that would work on Mars with little or no modification. The question is not whether bacterial life could survive on Mars. Earth bacteria have already proven they can under laboratory simulation of Martian conditions. The question is whether bacteria from 3i Atlas brought the appropriate genetic toolkit with them, and whether they landed in locations where survival was possible. If they did, then somewhere beneath the Martian surface, the survival equation is being solved in real time. Cells are dividing slowly but persistently. Deoxyribonucleic acid is mutating in response to new selective pressures. Natural selection is operating on timescales that make our impatience irrelevant. The great experiment of life adapting to a new world is underway, hidden from our current instruments but potentially laying the foundation for entire ecosystems that will develop over millions of years. What we are witnessing, if this scenario proves correct, is not just bacterial survival. It is the birth of Martian biology, shaped by terrestrial genes but optimized for an alien world. As we drift toward our next exploration, consider the profound implications of survival at these extremes. Life does not just endure impossible conditions, it transforms them into opportunities. What seems like the most hostile environment in the solar system might actually be the birthplace of Earth's first confirmed extraterrestrial colony, populated by descendants of bacteria that travelled farther than any Earth organism in history. The survival equation has been solved countless times in Earth's deep subsurface, in boiling springs, in nuclear reactors, in the vacuum of space itself. Now it may be operating on Mars, quietly converting the red planet from a sterile world into a living one. One bacterial generation at a time. Evolution by vertical inheritance, parent to offspring, generation after generation, is the story we learned in school, but it is not the complete story, and it's certainly not the most dramatic one. For bacteria, and possibly for life throughout the universe, horizontal gene transfer represents something far more revolutionary. The difference between waiting millions of years for beneficial mutations to arise randomly and acquiring those same beneficial traits in a single generation by copying them from a neighbour. The numbers tell a story that challenges everything we thought we knew about the pace of evolutionary change. Research has demonstrated that bacteria can exchange approximately 15% of their core genome in just 180 generations when two compatible lineages encounter each other. 180 generations sounds substantial for humans, where each generation spans decades. But for bacteria reproducing even once per week under hot conditions, that is less than 3.5 years. In more favourable environments where division occurs daily, we're talking about 6 months. This is not gradual accumulation of tiny changes over geological time. This is wholesale swapping of genetic modules, entire pathways and capabilities moving between organisms, as if bacteria were sharing software code in an open-source biological network. Imagine if humans could acquire the ability to see in the dark simply by standing next to someone who possessed that trait. That is essentially what bacteria accomplish through horizontal gene transfer. There are 3 primary mechanisms by which bacteria accomplish this genetic exchange, each operating under different conditions but all serving the same revolutionary purpose. Conjugation involves direct physical contact between cells, with one bacterium extending a structure called a pilus to another and literally pumping deoxyribonucleic acid through it like biological cable transfer. The transferred genetic material is typically carried on plasmids, small circular deoxyribonucleic acid molecules separate from the main chromosome. These plasmids often carry genes for environmental tolerance, metabolic capabilities, or resistance to toxic compounds. When a bacterium acquires a plasmid, it gains all those capabilities instantly, not through the slow accumulation of mutations, but through a single transformative event that can change its entire ecological role. Transformation represents a simpler but equally powerful mechanism. Bacteria can take up naked deoxyribonucleic acid directly from their environment, genetic material released when other cells die and disintegrate. This might seem like a random process, but many bacteria actively promote it by developing competence, a physiological state where their membranes become permeable to large molecules like deoxyribonucleic acid. They essentially become genetic scavengers, grabbing fragments from the surrounding medium and incorporating them into their own genomes through recombination. If those fragments happen to contain useful genes, those capabilities become part of the cell's permanent genetic repertoire. Transduction involves viruses as unwitting delivery vehicles. Bacteriophages, viruses that infect bacteria, sometimes accidentally package host deoxyribonucleic acid along with their own genetic material. When they infect the next bacterial cell, they inject that borrowed deoxyribonucleic acid. If the injected genes help the new host survive, natural selection favours bacteria that retain those genes. Phage-mediated gene transfer is common in aquatic environments on Earth. Whether phages exist on Mars or arrived aboard 3I Atlas with their bacterial hosts remains unknown, but if they do, they would accelerate genetic mixing beyond what direct cell contact could achieve. What makes horizontal gene transfer genuinely revolutionary for adaptation is how it navigates what biologists call fitness landscapes. In traditional Darwinian evolution, a population climbs gradually up a fitness peak through accumulated mutations. But imagine there is a higher peak separated by a valley of reduced fitness. The population would need to become less fit before it could become more fit, and natural selection actively prevents that kind of backward movement. Horizontal gene transfer solves this problem by allowing organisms to jump directly from one fitness peak to another, without crossing the valley of reduced survival. A bacterium struggling in one ecological niche can acquire genes that make it competitive in a completely different niche, all in one step. It is like having access to a biological teleportation network. Laboratory experiments have demonstrated this effect with startling clarity. Researchers evolved bacterial populations under controlled conditions, then introduced deoxyribonucleic acid from bacteria adapted to different environments. Populations with access to foreign deoxyribonucleic acid consistently adapt faster than populations relying solely on mutation, often by factors of 8 to 12 times. They explore more of the possible genetic solution space. They find adaptive solutions that mutation alone might take thousands of generations to discover. The effect becomes most pronounced when the environment is harsh or rapidly changing. Exactly the conditions bacteria from 3I Atlas would encounter on Mars. One particularly revealing study examined bacterial adaptation to antibiotic exposure, and the results have profound implications for our scenario. Populations were grown in environments without antibiotics, but with access to deoxyribonucleic acid from antibiotic-resistant strains. Over time, the non-resistant bacteria picked up resistance genes at low frequency. These genes provided no benefit in the antibiotic-free environment, so they remained rare and seemingly useless in the population. But when antibiotics were suddenly introduced, populations carrying those dormant resistance genes exploded in number, while populations without access to foreign deoxyribonucleic acid went extinct. Horizontal gene transfer did not just accelerate adaptation, it enabled survival under conditions that would otherwise be lethal. The genes were acquired before they were needed, sitting silent in bacterial genomes like evolutionary insurance policies, and waiting for environmental conditions that would make them valuable. This has profound implications for bacteria from 3I Atlas encountering Martian conditions. Even if the alien organisms are poorly adapted initially, even if they struggle to metabolize Martian chemistry or tolerate Martian temperatures, they can potentially acquire the necessary capabilities from native Martian bacteria, if any exist. Conversely, native Martian organisms could acquire novel capabilities from the newcomers. Both populations benefit. Both become more resilient through genetic cooperation. The rate of horizontal gene transfer depends on several critical factors, and understanding these factors helps us predict what might be happening in Martian subsurface environments right now. Physical proximity is essential. Bacteria need to be close enough for conjugation to occur, or for released deoxyribonucleic acid to reach competent cells before it degrades. Environmental deoxyribonucleic acid does not persist indefinitely. Enzymes called nucleases present even in harsh environments from the breakdown of dead cells, systematically destroy exposed genetic material over time. In Earth's soils, naked deoxyribonucleic acid typically persists for days to weeks before being completely degraded. But in Martian ice or frozen brine, where enzymatic activity is slowed by extreme cold, genetic material could remain intact for years or even centuries. Genetic compatibility represents another crucial factor. Not all deoxyribonucleic acid can be successfully integrated into any genome. Bacteria have evolved sophisticated molecular immune systems called restriction modification systems that cut up foreign deoxyribonucleic acid lacking the right molecular signatures. These systems protect bacteria from incorporating genetic material that might be harmful or incompatible. But these protective systems can be overcome if the transferred deoxyribonucleic acid provides sufficient survival advantage. Bacteria that fail to restrict beneficial foreign deoxyribonucleic acid will outcompete bacteria that do restrict it. Over time, populations become more permissive to gene transfer when the environment rewards genetic flexibility. The structure of bacterial genomes facilitates this kind of genetic mixing in ways that make it almost inevitable under the right conditions. Unlike eukaryotic organisms with tightly organized chromosomes, bacteria often have loosely arranged genomes peppered with insertion sequences and mobile genetic elements. These are stretches of deoxyribonucleic acid that can move around within the genome, or jump between different deoxyribonucleic acid molecules. They create natural recombination hotspots where foreign deoxyribonucleic acid can insert without disrupting essential functions. Once inserted, if the acquired genes prove beneficial, they are retained through natural selection. If they are harmful or useless, they are lost through deletion or outcompeted by cells that did not acquire them. One of the most powerful aspects of horizontal gene transfer is its ability to move entire functional modules simultaneously. On Earth, genes for related functions often cluster together in operons, groups of genes transcribed as a single unit. When an operon transfers between bacteria, the recipient gains a complete metabolic pathway or regulatory system all at once, not piece by piece over many generations. This is why antibiotic resistance can spread so rapidly through bacterial populations. A single plasmid might carry resistance genes for 5 different antibiotics, plus the regulatory elements that control their expression. One transfer event creates multi-drug resistance. If bacteria from 3i Atlas carry genes organized in similar operons, those complete functional units could transfer to Martian bacteria as intact modules. Consider the possibilities. A cluster of genes for radiation resistance, acquired as a complete package. A metabolic pathway for perchlorate reduction, transferred wholesale. A system for extracting phosphorus from specific mineral types, moved intact from one organism to another. Each transfer would represent a major adaptive leap, not an incremental improvement. This is evolution operating in fast-forward mode. Species-level changes occurring in the time it would normally take for a single beneficial mutation to spread through a population. There is also a fascinating feedback loop that accelerates the entire process. Horizontal gene transfer becomes more common in stressful environments because stress induces competence. When bacteria are starving, radiation damaged, or chemically poisoned, they are more likely to take up foreign deoxyribonucleic acid. It is a desperate biological strategy, a last resort before death. Most of the deoxyribonucleic acid they acquire is useless or harmful, adding to their burden rather than helping. But occasionally, purely by chance, they grab genes that solve their immediate problem. Those rare successes are what natural selection preserves and amplifies. Mars represents the definition of a stressful environment for any bacteria, whether native or introduced. Low nutrients, extreme cold, radiation, chemical toxicity. Every bacterium would be under constant stress. That stress would drive high rates of competence and transformation. Deoxyribonucleic acid release from dying cells would be continuous. Surviving cells would be constantly sampling that environmental genetic library. The rate of genetic exchange could be orders of magnitude higher than in the comfortable laboratory environments where most studies are conducted. There is another aspect that makes this scenario even more plausible. Gene transfer networks on Earth show strong ecological structure. Bacteria in the same habitat exchange genes more frequently than bacteria in different habitats, even if they are more closely related to organisms in different environments. The explanation is straightforward. Genes that help in one environment might be useless or harmful in another. Bacteria benefit most from acquiring genes from neighbours facing similar challenges. This means that even if bacteria from 3I Atlas are genetically distant from any native Martian life, if they end up in similar habitats facing similar problems, gene transfer between them becomes likely. Recent research has revealed that in natural bacterial communities, a significant fraction of successful gene transfers involve genes related to surface structures, nutrient transport, and stress response. These are exactly the categories that would be most valuable in Martian subsurface environments. Genes for modified membrane structures that function in high salt or low temperature, genes for transporters that scavenge trace nutrients, genes for repairing radiation damage or tolerating oxidative stress. These functional categories would be under intense selective pressure on Mars, making them the most likely candidates for horizontal transfer between any bacterial populations that manage to establish themselves. The long-term result of high rates of horizontal gene transfer is something that challenges our basic concepts of species and evolution. Instead of clear phylogenetic trees showing ancestry, bacterial populations become genetic mosaics, networks showing complex patterns of gene sharing between distantly related lineages. An organism's genome becomes a patchwork. Some genes inherited vertically from ancestors, others acquired horizontally from distant relatives, still others picked up from completely unrelated lineages that happen to solve similar problems. The concept of bacterial species becomes fluid, because genetic boundaries are so permeable. On Mars, this could mean that within a few thousand generations, you could not distinguish bacteria descended from 3I Atlas from bacteria descended from native Martian life, assuming any exists. Both lineages would have exchanged so many genes that their genomes would represent hybrids carrying the best solutions from multiple evolutionary histories. They would all be Martian bacteria at that point, uniquely adapted to their planet, carrying genetic information from multiple origins, but blended into new combinations that never existed anywhere else in the universe. The horizontal revolution is not just about faster adaptation, it is about expanding the realm of evolutionary possibility itself. It allows organisms to access genetic solutions that mutation alone might never discover. It enables rapid response to environmental change. It creates biological diversity without waiting for slow speciation processes. And most importantly for our story, it means that life, once introduced to a new world, does not have to reinvent every adaptation from scratch. Life can borrow, steal, and remix the genetic innovations of whatever other organisms it encounters, building something new from the combined wisdom of separate evolutionary histories. If bacteria from 3I Atlas are adapting to Mars right now, they are not doing it alone. They are part of a genetic network that might include native Martian organisms, previous deliveries from other comets, and possibly even Earth bacteria carried by spacecraft. The horizontal revolution is transforming Mars from a collection of isolated organisms struggling individually to survive into a cooperative biological network where genetic innovations spread rapidly through the entire community. It is evolution as we have never seen it before, not the slow grind of mutation and selection, but the rapid exchange of solutions between organisms facing common challenges, and somewhere beneath the Martian surface, in ice deposits and rock fractures, this revolution may be unfolding right now. 3I Atlas passed Mars at 29 million kilometers, but its closest approach to Earth occurred at a distance of 270 million kilometers, nearly 10 times more remote. Yet distance represents only one variable in the complex equation of microbial dispersal across the solar system. What matters more is the debris cloud itself, that vast expanding sphere of particles that the comet released as it approached perihelion and continues to shed as it moves away from our Sun. That cloud has grown large enough and diffuse enough that some fraction of it will intersect Earth's orbit, bringing with it the possibility of biological contamination from another star system. The mathematics are both humbling and concerning. 3I Atlas ejected between 100 and 800 grams of material every second for months during its solar approach. Let us be conservative and estimate an average of 400 grams per second sustained over 120 days around perihelion. That represents over 4 million kilograms of dust and ice particles, released into trajectories that now crisscross the inner solar system. Most of that material will never approach Earth. It will drift outward with the comet or fall inward toward the Sun, eventually finding destinations far from our world. But statistical mechanics tells us with certainty that some fraction, however infinitesimally small, will end up on trajectories that bring them to our planet over the coming months and years. Earth already intercepts roughly 100 metric tons of extraterrestrial material every day, most of it dust from comets and asteroids, particles so small they drift down through our atmosphere without creating visible meteors. We have found these particles everywhere we have looked, in ice cores preserving atmospheric history, in deep-sea sediments accumulating on ocean floors, in high-altitude atmospheric samples collected by research aircraft. They are ubiquitous and mostly ignored, because they are typically inert, just minerals and occasional organic molecules left over from the solar system's formation. But what if some of the particles settling onto Earth over the next few years are not ancient debris from our own solar system's youth? What if they are fresh material ejected from 3I Atlas, carrying biological cargo from another star system? The implications are profound and deeply unsettling, challenging our assumptions about planetary isolation and biological security. Earth presents a radically different reception environment than Mars, where the red planet offers a whisper-thin atmosphere that barely interferes with incoming particles. Our world presents multiple layers of defense against cosmic contamination. The first barrier is atmospheric entry itself, and even particles in the micron range experience significant heating as they collide with air molecules during their descent. For larger particles, to those in the millimeter range, the heating can be extreme enough to create plasma around the incoming object. The outside surface chars and ablates away, in a process that would sterilize any biological material exposed to the thermal shock. But studies of micrometeorites recovered from Earth's surface reveal something intriguing. While the exteriors are melted and oxidized beyond recognition, the cores often contain pristine minerals and even organic compounds that never reach destructive temperatures. If a particle from 3I Atlas containing bacterial spores enters Earth's atmosphere, the microorganisms on the surface would be sterilized instantly. But spores embedded deeper inside, particularly within ice-cemented dust aggregates, might survive the entry process. The ice would sublimate during atmospheric passage, providing cooling through phase change. By the time the particle slows to terminal velocity in the lower atmosphere, it would be cold again, and any surviving spores would arrive on Earth's surface dormant but potentially viable. Where these particles land determines everything that follows in their biological story. If they settle into the ocean, they enter the most competitive environment on the planet. Marine bacteria are extraordinarily diverse, aggressive, and territorial. They have evolved sophisticated chemical warfare capabilities, producing antimicrobial compounds that exclude competitors. They form biofilms that physically block newcomers from accessing nutrients. After billions of years of evolution in Earth's oceans, they have created biological fortresses that newcomers struggle to penetrate. An alien bacterium, even one with sophisticated survival mechanisms developed during interstellar travel, would face overwhelming competition from organisms perfectly adapted to marine conditions. The newcomer might survive for hours or days, but establishing a reproducing population would be nearly impossible against such entrenched opposition. If the particles land on exposed rock or desert surfaces, ultraviolet radiation becomes the immediate threat. While Earth's ozone layer blocks the harshest ultraviolet wavelengths that bombard Mars, enough penetrates to sterilize exposed surfaces within hours. Bacteria deposited on sunlit rock or sand would face deoxyribonucleic acid damage faster than their repair mechanisms could compensate, leading to rapid death. But if particles from 3I Atlas land somewhere protected, in soil beneath vegetation canopies, in the shadows of rocks or buildings, in the perpetually dark deep ocean, or in polar regions where ice accumulates, then they might have genuine opportunities for revival and establishment. The problem they would face is competition on a scale that makes Martian conditions seem benevolent. Earth's soil contains between 10 million and 10 billion bacterial cells per gram. Depending on location and conditions, that is not a typographical error. We're talking about 10 to the 7th through 10 to the 10th, organisms competing for resources in every handful of dirt you might pick up. These terrestrial bacteria have specialized into countless ecological roles over billions of years. There are decomposer specialists for breaking down every type of organic matter imaginable. There are predatory bacteria that actively hunt other microorganisms. There are chemical warfare specialists that produce toxins specifically designed to kill competitors. It is biological warfare at the microscopic scale, conducted with a sophistication that makes human conflict seem crude. For an alien bacterium to establish itself in such an environment, it would need extraordinary advantages. It would need to metabolize compounds that Earth bacteria cannot use, or metabolize common substrates much more efficiently, or tolerate conditions harsh enough to exclude native organisms. The first two scenarios are unlikely, because Earth bacteria have had 4 billion years to optimize for Earth chemistry. The third possibility, finding unoccupied ecological niches, offers the most realistic pathway to successful colonization. High radiation environments might provide one such niche. While Earth hosts radiation-resistant bacteria, they're relatively uncommon because most of our planet's surface is shielded from intense radiation by atmosphere, water, or rock. A bacterium from 3I Atlas that evolved during billions of years of cosmic ray exposure might possess radiation resistance capabilities that exceed anything terrestrial evolution has produced. If such organisms landed near nuclear facilities, in uranium-rich geological formations, or in other high radiation environments, they might find ecological space where they could survive without direct competition from native species. They would not dominate these environments immediately, but they might establish small populations that could serve as founding colonies for further expansion. Extreme pH environments represent another possibility. Earth hosts acidophiles that thrive at pH 1.5, more acidic than battery acid. It also has alkalophiles that flourish at pH 13, as caustic as industrial bleach. But the specific metabolic strategies these organisms employ evolved here, optimized for Earth's particular chemical conditions. A bacterium from 3I Atlas might have developed different approaches to handling pH extremes, strategies that work better in specific environmental contexts that Earth bacteria find marginal. Those contexts could provide niches where newcomers survive alongside natives, rather than being outcompeted entirely. But the real concern from a planetary protection perspective is not direct competition between alien and terrestrial bacteria. It is the possibility of genetic exchange between the two populations. This is where horizontal gene transfer transforms from an accelerator of adaptation into a potential vector for biological disruption. If organisms from 3I Atlas carry genes that provide novel capabilities, Earth bacteria that acquire those genes through transformation, conjugation, or transduction gain advantages they never possessed before. Most of the time, this process is neutral or beneficial. New metabolic pathways, improved stress tolerance, enhanced nutrient utilization. These represent genetic innovations that could help Earth's biosphere adapt to changing environmental conditions. But there are darker possibilities that keep astrobiologists awake at night. Imagine an alien bacterium carrying genes for toxin production, compounds that affect eukaryotic cells in ways Earth organisms have never encountered. Native bacteria that acquire those genes could become pathogenic to plants, animals, or humans, even if the original alien organism was completely harmless. Or consider genes that confer extreme environmental tolerance, enabling bacteria to survive in places they could never colonize before. Industrial facilities designed to be sterile could become contaminated. Medical environments could harbor resistant organisms. Even clean rooms built to exclude all biological contamination could become breeding grounds for hybrid bacteria carrying alien survival genes. The probability of any specific harmful outcome is low, but when you are dealing with the astronomical numbers involved in cometary debris, billions of particles over extended time periods, even extremely low probability events enter the realm of possibility. Most particles from 3I Atlas that reach Earth will be sterilized during entry or upon landing. Most survivors will be unable to compete with native organisms. Most genetic exchanges will transfer capabilities that are redundant with what Earth bacteria already possess. But most is not all. And the small fraction of exceptions could have consequences that ripple through ecosystems in unpredictable ways. There is historical precedent for concern here. Earth bacteria have been introduced to new continents multiple times throughout human history, often with devastating ecological consequences. Diseases that evolved in isolation suddenly encounter populations with no immunity. Invasive species that lack natural predators in their new environment proliferate beyond control. The biological equivalent of ecological disasters can result from introducing the wrong organism into an unprepared ecosystem. Now extrapolate that scenario from continental to interplanetary scale. Instead of moving organisms between regions on the same world, we are potentially talking about genetic exchange between completely different evolutionary histories, shaped by different planetary environments over billions of years. The counter-argument is that Earth has been receiving extraterrestrial material throughout its entire geological history. Meteorites fall constantly. Comets deposit material regularly during their periodic returns. If alien bacteria were going to cause problems, would they not have done so already? This reasoning suggests either that viable alien bacteria have never reached Earth in sufficient numbers to establish themselves, or that they have, but we have not noticed because the effects were too subtle to detect. There is a third possibility that is both reassuring and unsettling. Maybe alien bacteria have successfully colonized Earth multiple times throughout geological history. Maybe some fraction of our planet's bacterial diversity does not descend from a single origin of life here. Maybe some lineages carry genetic information from other star systems, information that has become so thoroughly integrated with terrestrial genes through billions of years of horizontal transfer that we cannot distinguish the original sources. If true, then life on Earth represents a hybrid system carrying the genetic legacy of multiple worlds, with each new arrival adding to an already cosmopolitan collection. Adding material from 3I Atlas would not fundamentally change the nature of terrestrial life. It would simply contribute new genetic possibilities to an already diverse library. But 3I Atlas differs from previous interstellar visitors in one crucial respect. We are aware of it. We are tracking it. We know it passed close to Mars, and that its debris cloud intersects Earth's orbit. If there are biological consequences, if alien bacteria do establish themselves in terrestrial environments, we might actually be able to detect the changes this time. Not immediately, perhaps not for years or decades, but eventually we should see novel genetic sequences appearing in bacterial populations. We would observe new metabolic capabilities spreading through microbial communities. We would detect evidence in the genomic record of recent horizontal gene transfer events, from sources that do not match any known terrestrial lineages. Searching for these signatures requires systematic genomic sequencing of bacterial populations from diverse environments, looking specifically for anomalies that suggest recent genetic introductions, genes that cluster together in ways that do not match standard phylogenetic relationships, metabolic pathways that use enzymes slightly different from terrestrial equivalents, proteins with amino acid sequences optimized for conditions that do not exist in the environments where they are found. These would be subtle clues, easy to miss without deliberate investigation. But they would constitute smoking guns if detected, direct evidence of recent genetic exchange with extraterrestrial sources. There is also the possibility of more obvious effects if alien bacteria carrying novel genes establish themselves in specific environments. Unusual algal blooms in water bodies that do not respond to typical controls, shifts in soil chemistry that cannot be explained by known biological processes, changes in the dominant bacterial species in particular ecological niches. Environmental monitoring programs might detect these signals without initially recognizing their significance. A slight increase in a rare bacterial species here, an unexplained change in nutrient cycling rates there, individually meaningless, but collectively suggesting that something new has entered the system and is beginning to influence local ecology. The timeline for such effects to become apparent would span years to decades. Bacterial evolution operates on generational scales, and even with rapid reproduction, it takes time for new genes to spread through populations, for natural selection to act on novel traits, for measurable changes to accumulate in environmental systems. We would not see sudden dramatic transformations like science fiction scenarios of alien invasion. We would see gradual shifts, trends that only become clear when comparing data from before and after 3I Atlas's passage through the inner solar system. This is where citizen science initiatives could play a crucial role in detection efforts. Amateur microbiologists collecting and sequencing bacteria from local environments, distributed monitoring projects tracking changes in microbial communities across wide geographic areas, databases where researchers can upload sequence data for automated analysis, looking for phylogenetic anomalies. The more eyes watching bacterial populations, the more likely we are to catch something unusual if it appears. But the reality is that we probably will not know whether bacteria from 3I Atlas successfully colonized Earth unless they cause obvious problems, or unless we get extraordinarily lucky with genetic sequencing efforts. The microbial world is vast and largely unexplored. We have characterized perhaps 1% of bacterial species on Earth, leaving 99% unknown, their genes unread, their capabilities uncharacterized. Against that background of ignorance, detecting the introduction of alien organisms resembles finding specific needles in a planet-sized haystack. What we can say with confidence is that Earth's uninvited guests, if they're here, face the same fundamental challenges that confront any invasive species. They must survive initial conditions hostile to their biology. They must find suitable resources in environments already occupied by superbly adapted competitors. They must reproduce faster than they die, while avoiding the countless threats that native organisms have learned to counter. It is not an impossible challenge, but it is extraordinarily difficult, which is probably beneficial for planetary security, because a universe where biological contamination between worlds was easy would be one where planet-scale ecological disasters were inevitable and frequent. The particles from 3I Atlas that reach Earth represent a natural experiment in interplanetary biology. Most will fail to establish reproducing populations, but some might succeed, adding their genetic innovations to Earth's already staggering biological diversity. Whether such additions prove beneficial, neutral, or harmful will depend on details we will not know until the experiment concludes. For now, we can only watch and wait, monitoring bacterial populations for signs of change, sequencing genomes for evidence of recent horizontal transfers, and trying to understand how life behaves when the boundaries between worlds become permeable to biological exchange. The cosmic seeds from 3I Atlas may already be taking root in terrestrial soil, beginning the slow process of integration with Earth's biosphere. Or they may have failed entirely, leaving no trace of their interstellar journey. Either way, they represent something profound, life's endless drive to explore, to spread, to test the limits of what is possible in a universe where biology recognizes no boundaries that physics does not enforce. As we drift toward our final exploration, consider this. You may be breathing air that contains particles from 3I Atlas right now. The bacteria in your microbiome may already be encountering genetic material from another star system. The boundary between terrestrial and extraterrestrial life may be far more porous than we ever imagined, making each of us unwitting participants in the greatest biological experiment ever conducted. Project forward with me now. Not years, but millennia. Not thousands of years, but millions. If bacteria from 3I Atlas successfully colonized Mars's subsurface, what would that biosphere look like as it matures and evolves? This is not speculation for the sake of wonder. It is applying what we understand about bacterial evolution, ecology, and planetary science to envision plausible outcomes that could be unfolding beneath our feet and above our heads right now. The starting point is a sparse population of microorganisms distributed unevenly through Martian subsurface ice deposits, rock fractures, and possibly the vast aquifer systems that may exist beneath the polar caps. These organisms exist at the absolute limits of what biology can tolerate, where growth rates are so slow that reproduction might occur once per year, once per decade, or even once per century, depending on how cold and nutrient-poor their specific microenvironment proves to be. At these glacial rates of reproduction, even 1 million years represents only 10,000 to 100,000 generations, sufficient time for significant evolutionary change in bacterial populations, but not enough for complete biological transformation. The first major evolutionary pressure these populations would face is optimization for extreme cold tolerance that goes beyond anything Earth has produced. Earth's most cold-adapted bacteria, the psychrophills, can grow at temperatures down to minus 17 degrees Celsius, below that threshold. Metabolism essentially ceases in pure water, but Martian subsurface environments do not contain pure water. They harbour brines, salt-saturated solutions that remain liquid at much lower temperatures. Bacteria capable of functioning in these hypersaline, supercooled environments would possess enormous advantages over any competitors. Evolution toward extreme cold adaptation involves coordinated changes across multiple biological systems. Enzymes need to remain flexible at temperatures where most proteins become rigid. Cellular membranes must stay fluid when ice crystals would normally freeze them solid. Deoxyribonucleic acid repair mechanisms need to function despite chemical reactions proceeding millions of times slower than at moderate temperatures. Each of these adaptations requires specific genetic modifications, multiple mutations coordinating to shift the organism's entire physiology toward cold tolerance. If bacteria from 3I Atlas brought genes for extreme cold adaptation from their home system, optimized during billions of years of interstellar travel, those capabilities would spread rapidly through Martian bacterial populations via horizontal gene transfer. Within a few thousand generations, we would expect to see cold tolerance genes present in most or all bacterial lineages that survived the initial colonization. The population would become collectively more cold tolerant, not through independent evolution of the same solutions, but through sharing the most effective genetic innovations discovered by any member of the community. The second major evolutionary pressure would be radiation management that surpasses anything terrestrial life has encountered. Even in Martian subsurface environments, bacteria face constant bombardment from cosmic rays and radioactive decay of surrounding mineral deposits. Over millions of years, accumulated radiation damage would be staggering, requiring deoxyribonucleic acid repair mechanisms of extraordinary sophistication. If bacteria from 3I Atlas carried superior radiation resistance genes developed during their journey between stars, those genetic tools would become valuable currency in the emerging Martian gene pool. But there is an important twist in this evolutionary scenario. Radiation creates reactive oxygen species, highly damaging molecules that attack cellular components. On Earth, aerobic bacteria evolved elaborate systems for managing oxidative stress because they live in an oxygen-rich atmosphere. Mars' atmosphere consists almost entirely of carbon dioxide, with virtually no free oxygen. Martian bacteria would not face atmospheric oxidative stress, only radiation-induced reactive species. They might evolve completely different approaches to managing oxidative damage, optimized for low oxygen, high radiation conditions rather than the high oxygen, low radiation environment that shaped terrestrial bacteria. The third evolutionary pressure would be nutrient scavenging efficiency in an environment where every essential element is scarce. Nitrogen exists only in trace amounts. Phosphorus remains locked in slowly dissolving minerals. Essential trace elements like iron, zinc, and molybdenum are present but not necessarily accessible to biological systems. Bacteria capable of efficiently extracting nutrients from their environment would consistently outcompete less efficient scavengers, driving evolution toward improved uptake systems, more effective mineral-dissolving enzymes, and sophisticated nutrient recycling mechanisms. Over millions of years, these selection pressures would sculpt Martian bacterial communities into something distinctly non-terrestrial. The organisms would still be recognizably bacterial, still using deoxyribonucleic acid, proteins, and lipid membranes, but their specific adaptations would be unique to Mars. They would be optimized for hypersaline brines, intense radiation, and extreme nutrient limitation in ways Earth bacteria never needed to develop. The ecological structure would be elegantly simple compared to Earth's baroque complexity. No photosynthesis, because there is no light in subsurface environments. No oxygen-based respiration, because there is no atmospheric oxygen. The entire ecosystem would operate on chemosynthesis, bacteria extracting energy from chemical reactions between minerals and dissolved gases. Hydrogen oxidation would likely serve as the primary energy source, with carbon dioxide reduction as the main carbon-fixing pathway. Sulfur and iron cycling would connect different metabolic guilds in intricate biochemical networks, but simplicity does not imply stasis. Within these constraints, diversification could occur as different bacterial lineages specialize for different chemical reactions. Some might focus on hydrogen oxidation coupled to sulfate reduction. Others might specialize in iron reduction or perchlorate reduction. Still others might function as decomposers, breaking down organic molecules produced by the primary producers. These different metabolic strategies would create ecological structure, different species occupying distinct biogeochemical niches in the subsurface ecosystem. Spatial structure would emerge as well, driven by the heterogeneous nature of Mars's subsurface. Bacteria near geothermal features where subsurface heat maintains liquid water at higher temperatures would form different communities than bacteria in the frigid polar ice. Organisms in porous sandstone would face different challenges than those in dense basalt. Communities in sulfate-rich evaporate deposits would differ from those in iron-rich volcanic rocks. Each environment would select for slightly different traits, creating geographic variation across Mars' subsurface. Different bacterial provinces adapted to local conditions but connected through the slow movement of water and the exchange of genetic material. The most intriguing possibility is the potential emergence of multicellularity in response to Mars' extreme nutrient limitation. Earth bacteria occasionally form multicellular structures like biofilms and fruiting bodies that allow collective behavior, resource sharing, and division of labor. In extremely nutrient-poor environments like Earth's deep subsurface, bacteria sometimes form elongated filaments connecting individual cells, possibly to facilitate nutrient distribution across large distances. Martian bacteria facing similar extreme nutrient scarcity might evolve even more elaborate multicellular networks. Imagine interconnected bacterial filaments spreading through rock fractures, linking cells in warm, nutrient-rich zones with cells in cold, nutrient-poor regions. The network as a whole could be more resilient than individual cells, buffering against local environmental fluctuations and distributing resources where they are needed most. This would not represent multicellularity in the animal or plant sense, but it would constitute a level of biological organization beyond individual cells responding independently to their environment. A colonial intelligence distributed through Martian rock, invisible to surface observers, but potentially spanning continental scales. The temporal framework for these developments remains uncertain, but we can make educated estimates based on terrestrial analogies. On Earth, bacterial ecosystems can reach dynamic equilibrium relatively quickly when resources are abundant and conditions favourable. But Mars offers neither abundance nor favourable conditions. Everything happens slower there. Cell division occurs at glacial pace. Natural selection acts more weakly because survival margins are so narrow. Opportunities for horizontal gene transfer are less frequent because population densities remain low. A reasonable estimate might place significant diversification in the millions to tens of millions of years time frame, with truly novel adaptations not present in founding populations requiring perhaps 100 million years to evolve. These are lengthy periods by human standards, but brief in geological terms. Mars has existed for 4.5 billion years. If life has been attempting to establish itself repeatedly through cometary deliveries over that entire span, there has been ample time for sophisticated bacterial ecosystems to develop, even under harsh constraints. This raises a profound possibility. What if sophisticated Martian bacterial ecosystems already exist? What if the 3i Atlas delivery is not the first successful colonization event, but merely the latest in a series stretching back billions of years? Native Martian life might already be diverse, already well adapted, already occupying available niches in subsurface environments. In that scenario, bacteria from 3i Atlas would be immigrants entering an established ecosystem rather than pioneers colonizing an empty world. The implications for detection are sobering. If Martian life exists but remains confined to deep subsurface environments, our current exploration methods will miss it entirely. Surface rovers can only scratch the uppermost layers of soil. Planned drilling missions target depths measured in meters, not kilometers. Orbital instruments can detect surface compositions but cannot penetrate to the subsurface ice deposits, where life would most likely persist. To find deep biospheres, we need dedicated missions with deep drilling capabilities, subsurface imaging systems designed to detect biological signatures, and sample return missions targeting the most promising subsurface habitats. Several locations on Mars deserve priority attention in such searches. The region beneath the southern polar ice cap where liquid water has been detected by orbital radar. The deep aquifer systems that may connect to ancient river valley networks. Areas showing evidence of recent fluid flow in gullies and recurrent slope lineae, geothermally active regions where volcanic heat might maintain liquid water conditions. Each environment could harbour bacterial communities adapted to local conditions, whether native or introduced through cometary delivery. The search strategy should focus on biosignatures that persist even when organisms themselves are rare or dormant. Lipid molecules from bacterial membranes can preserve in rocks for billions of years. Isotopic signatures from metabolic processes remain detectable long after organisms die. Minerals like magnetite or sulfides produced through bacterial metabolism can indicate biological activity. Deoxyribonucleic acid fragments, if protected in ice or mineral matrices, might persist long enough for detection even without viable cells. But the ultimate goal transcends simply finding evidence of life. It involves understanding entire biospheres as integrated systems. What organisms are present? How do they relate to each other and to Earth life? What metabolic strategies do they employ? How do they interact with their physical environment? A mature Martian biosphere would represent a complex web of metabolic interdependencies, with different species relying on each other's waste products and creating feedback loops that stabilize the system against environmental perturbations. On Earth, detailed genetic analysis has revealed that apparently simple microbial communities actually consist of hundreds or thousands of distinct lineages interacting in intricate ways. Martian bacterial communities might display similar hidden complexity. Surface observations might reveal nothing but sterile rock, while meters below, rich ecosystems thrive invisibly. The future Martian biosphere, whether developing from 3i Atlas colonization already established through previous deliveries, would represent something unique in our current understanding of biology. It would be life adapted to conditions at the absolute edge of habitability, surviving through strategies we can barely imagine, creating ecosystem function on timescales that make human lifespans irrelevant. It would serve as a laboratory for understanding how far biology can be pushed, what universal principles govern life regardless of environment, and what diversity becomes possible within the constraints of physics and chemistry. And someday, when humans establish permanent presence on Mars, we will face crucial decisions about how to interact with this biosphere. Do we protect it, maintaining pristine scientific preserves for study? Do we ignore it, focusing on surface habitats where it does not exist? Do we try to enhance it, deliberately introducing organisms that might help terraform the planet? These are not merely scientific questions. They are ethical questions about our responsibilities to other forms of life, and about what we want Mars to become. The future Martian biosphere might be invisible now, but its foundations could be settling into place as debris from 3i Atlas drifts down through the thin atmosphere and begins the patient process of adaptation. In 1 million years, Mars might host thriving subsurface ecosystems cycling elements and processing minerals in ways that transform planetary chemistry. In 10 million years, it might teem with bacterial life so thoroughly adapted to Martian conditions that they could not survive anywhere else in the solar system. and all of it might trace back to an interstellar comet that happened to pass by, carrying passengers from distant stars. But our story extends beyond Mars to encompass Earth itself and the broader implications of what we have explored. If life on Earth contains genetic information from other star systems, delivered by comets over billions of years of planetary history, then our origin story becomes more complex and more beautiful than we previously understood. We are not purely products of terrestrial evolution. We are hybrids carrying biological heritage from multiple worlds, blended together over cosmic timescales through the patient work of cometary messengers. Every organism on Earth, including humans, might contain genes that originated elsewhere, so thoroughly integrated into our biology that we cannot distinguish them from local innovations. This challenges the narrative of human uniqueness and terrestrial isolation. We like to think of ourselves as special, the pinnacle of Earth's evolutionary experiment, but if Earth's biosphere has been regularly supplemented by genetic material from other systems, then we are not pure products of local development. We are participants in a galactic ecosystem, one world among many contributing to, and drawing from, a shared pool of biological information distributed throughout the galaxy. Some find this idea unsettling because it undermines the sense that Earth is our unique home, that we emerge from this specific place through an unrepeatable sequence of events. But I find it profoundly beautiful. It means we are connected to the larger universe not just through physics and chemistry, but through the actual genetic instructions that built us. We are not alone in cosmic isolation. We are participants in an exchange of life that has been ongoing since stars first formed planets capable of nurturing biology. This perspective transforms how we think about our place in the universe and our responsibilities as we expand into space. Current planetary protection protocols assume Mars might host native life, but those assumptions are theoretical, designed to prevent contamination of a potentially living world. If 3I Atlas delivered viable bacteria just months ago, then we are not protecting a hypothetical biosphere anymore. We are protecting an actual biosphere in the early stages of establishment. This creates ethical dilemmas without precedent. Do we have the right to interfere with bacterial colonization attempts? Should we help by deliberately introducing terrestrial organisms that might share useful genes? Should we hinder the process by sterilizing areas where we plan human activities? Or should we simply observe, maintaining distance and letting natural processes proceed? There is no consensus because we have never faced these questions outside theoretical discussions. Every previous conversation about planetary protection has been hypothetical. What we would do if we found life. Now we might be in a situation where life was recently introduced and is potentially establishing itself in exactly the environments we are planning to explore intensively. The decisions we make over the next few decades could determine whether Mars develops a robust biosphere or remains essentially sterile. If particles from 3I Atlas also reached Earth, those organisms are here now, probably struggling in marginal environments, unable to compete with native life. But possibly some found niches where they can survive. Possibly they are exchanging genes with terrestrial bacteria. Possibly we are already coexisting with organisms carrying genetic information from another star system. This is not an invasion scenario. It is not alien conquest. Most horizontal gene transfer events are neutral or beneficial, contributing capabilities that help organisms adapt to changing conditions. But we should monitor for unusual changes in bacterial populations, especially in extreme environments where novel organisms would be most likely to establish themselves. The monitoring does not require new infrastructure. We have environmental genomic sequencing programs, citizen science initiatives, and databases tracking bacterial diversity. What we need is awareness to look for specific signatures, genes that do not fit standard phylogenetic patterns, metabolic capabilities appearing suddenly in populations that lack them before, clusters of related sequences appearing simultaneously in distant locations. If we find evidence of colonization from 3I Atlas, the response should be measured study, rather than panic. Most likely, any successful colonist will prove harmless, just another set of bacterial species joining Earth's enormous diversity. The implications for our future as a spacefaring civilization are equally important. If panspermia operates naturally, if life spreads between worlds via comets and asteroids, then perfect planetary protection becomes impossible. We can minimize contamination, but we cannot prevent all biological exchange because nature has been conducting such exchanges for billions of years. This does not justify recklessness, but it suggests our approach should be pragmatic rather than absolutist. Protect against massive contamination that could overwhelm existing biospheres. Prevent introduction of organisms specifically designed to outcompete native life, but accept that small-scale exchanges are natural and have occurred throughout solar system history. Our spacecraft might carry thousands of bacterial spores, but a single comet can carry trillions. What we should focus on is understanding rather than just preventing. If we are going to be a spacefaring civilization, we need to understand how biology behaves in space, how organisms adapt to different planetary environments, and how ecosystems develop under non-terrestrial conditions. Mars will likely become a test case for these questions as we establish permanent human presence there. We will inevitably introduce Earth organisms, whether intentionally or accidentally. Bacteria will escape from habitats. Plants will release seeds and spores. Water recycling systems will leak microorganisms. Over decades and centuries, Mars will become contaminated with terrestrial life. The question is not whether this will happen, it is whether we manage it intelligently. Intelligent management means preserving biological reserves on Mars, areas where we do not go and do not contaminate, allowing continued searches for native or naturally introduced life. It means careful monitoring of how introduced organisms behave under Martian conditions and whether they spread beyond our settlements. The final implication concerns perspective and meaning. The story we have explored, bacteria from 3I Atlas potentially seeding Mars and Earth with life from another star, represents just one example of a process that is probably ubiquitous throughout the galaxy. Every star system with comets potentially exchanges life with every other system. Over billions of years, this creates a web of biological connections spanning vast distances. Life in the galaxy might not consist of isolated islands developing independently. It might be more like an archipelago, with occasional connections allowing exchange of species and genetic innovations. Some worlds might be sources, pumping out organisms that colonize other planets. Some might be sinks, receiving more than they contribute. Most, like Earth, are probably both simultaneously. This means that when we search for life elsewhere, we should expect biochemistry similar to ours, not just because similar chemistry leads to similar solutions, but because we might literally share genetic heritage with life on other worlds. The universal genetic code might be universal, not because it is the only possible solution, but because it's spread between worlds through panspermia. It also means we are not alone, even if we never encounter intelligent aliens. We are connected biologically to other worlds through bacteria we carry, genes we have inherited from comets, and organisms we will inevitably spread as we explore space. Every human contains bacteria descended from lineages that might trace back to other star systems. The 3I Atlas scenario is not just about whether a comet delivered bacteria to Mars and Earth. It is about recognizing that we are part of a living galaxy, where biological information flows between worlds just as matter and energy do, and where life's abilities to adapt and spread makes it a cosmic phenomenon rather than a planetary curiosity. We emerge from this process. We participate in it. As we expand into space, we become agents of it, deliberately and accidentally spreading Earth's biology to new worlds, just as other worlds may have contributed to our development. What this requires is that we think bigger, not just about life on Earth or Mars, but about life as a galactic process requiring ethics and policies appropriate for a species that is both beneficiary and agent of panspermia. We have travelled together through a hypothesis, following bacteria from an interstellar comet as they potentially seed two worlds with life from another star system. We have explored survival at extremes, evolution in fast forward, and the patient mechanisms by which life spreads between worlds over cosmic time. The bacteria from 3I Atlas, if they exist and if they survived, accomplished something we have not yet achieved: crossing interstellar space, surviving conditions that would destroy most of our technology, and adapting to alien worlds. These are achievements that deserve respect, even if the organisms involved are microscopic and non-conscious. They represent life's incredible resilience, its drive to persist against overwhelming odds, and its ability to spread wherever conditions permit. When we eventually drill into Mars' subsurface ice and find bacterial communities thriving there, communities that might trace ancestry to 3I Atlas or other cometary deliveries, we will be meeting distant relatives, not relatives through recent common ancestry, but relatives as fellow travellers in the cosmic dispersal of life. We're all expressions of the same fundamental phenomenon when you consider it deeply enough. Complex arrangements of matter organised by information that has flowed between worlds for billions of years. The story of 3I Atlas and its potential bacterial cargo continues to unfold. Some organisms might succeed spectacularly, founding lineages that persist for millions of years. Most will probably fail, joining countless other attempts at interstellar colonization that could not quite bridge the gap between worlds. But succeed or fail, they're part of something magnificent. The great experiment of life testing every world, finding every niche, persisting wherever persistence becomes possible, and we are part of that experiment too. What we do next matters not just for us, but for the future of life itself. So when you look up at the night sky and think about 3I Atlas somewhere out there, moving away from us back toward the stars, remember that it might have left something behind. Invisible passengers, frozen bacteria beginning the long process of adaptation to worlds that are not their home. They are out there now, in Martian ice or Earth's soil, quietly surviving or quietly dying, writing the next chapter in a story that includes you. Your microbiome carries bacteria that might have travelled between stars. Your genes might include sequences inherited from organisms that arrived on Earth through Panspermia. You're not just human, you're an ecosystem walking a collection of Earth's bacterial heritage, carrying within yourself the biological legacy of cosmic exchanges stretching back billions of years. The bacteria from 3I Atlas, if they made it here, are just the latest addition to a collection that has been accumulating since Earth first formed. We have wandered through cosmic time and biological space, exploring mysteries that connect worlds across impossible distances. The questions remain, as they should. Perhaps that is fitting. Perhaps the questions themselves are what matter most. For now, let this thought sit with you as you drift towards sleep. The universe is more alive than we ever dared imagine, and you are part of that life, carrying within yourself the patient work of billions of years of cosmic biology. The wandering messengers have delivered their gifts. What grows from those gifts, on Mars, on Earth, in the spaces between worlds, is still being written. Wherever you are, I hope you find rest in that vast living cosmos that connects us all. Thank you for wandering with me tonight through the silent spaces where life travels between stars,